Okay, this is your quarterly one BioSci review. One of the first things we started talking about this year was the scientific method. And we distinguished book science from real science. In book science, if you recall the steps of the scientific method, people often have excellent dinner conversation, problem, observations, which occurs before the hypothesis, an experiment you conduct, and data you collect separate from observations before the hypothesis, and you form a conclusion based on your data. If we take the experiment and recall how that is to be done, an experiment normally consists of a control group and a variable group, or experimental group. I believe we called it an experimental group. And the problem we had posed in class with the example we had down on the board was why a tree loses its leaves. So if your hypothesis is something along the lines of a tree loses its leaves because the weather gets colder, then this is the one that would have the colder temps. The control group would be without the hypothesis. It would have higher temps. And then recall that this is the only factor that will differ in your two groups. Every other factor that can be measured here. So we talked about, if I have an oak tree here, I need an oak tree here. If I'm in New Jersey here, I need to be in New Jersey here. The age of the tree, the amount of precipitation, 1 inch of rain, per week, 1 inch per week needs to be identical. So this tells you that if something happens with a differential result, whereas that this tree loses 80% of its leaves and this tree loses 50% of the leaves, well, the only thing that could have caused that, since all these factors were the same, was the one factor that varies. We spent some time and are still in a little bit of a hierarchy of classification of matter. At the bottom end or smallest end are atoms. Atoms make molecules. Molecules, when put together, make cells. Cells make something called tissues, like muscle and nerve. Tissues make organs, and organs make systems, which make up you, the organism. In this hierarchy, the first level, which you would say is alive, is the cellular level. Below here, you could have things like plastic and steel and water, which are obviously not alive. If we are discussing atoms, recall that atoms consist of a nucleus where proteins are contained and neutrons, and then electrons are circling in an orbit around that nucleus. Recall that protons are positively charged, neutrons have no charge, and electrons are negatively charged. Occasionally atoms hook together and form bonds with one another. And the one molecule, which you should have some familiarity with, and its bonds is water. Water is H2O. In a bond between oxygen and hydrogen, the electrons are shared. This is called a covalent bond. The other one is the ionic bond, in which electrons are transferred, but which you find in most molecules dealing with living things are covalent bonds. The covalent bond in water is a little bit different. What we call a polar covalent bond. So what do we know about poles? North pole and south pole are opposites. So what we have in water is oxygen, which is an electron hog, pulls the electrons in this shared bond closer to it and making these electrons further from hydrogen. This makes oxygen a little bit negative and hydrogen a little bit positive. So we have, if you will, the South Pole and the North Pole here. A lot of water's unique characteristics. So we discussed water being sticky. So what happens is a positive hydrogen is going to attract a negative oxygen and pull it along with it, creating things like surface tension in water, allowing a tree to pull water up its stem, up to the leaves. This polarity also allows water to dissolve certain substances. So if I have a beaker of water and I put in something that is also polar, like sugar, which also has a positive end and a negative end, or salt, which also has a positive end and a negative end, all the positives and negatives will attract each other, opposites attract, and you get this mixture of solute and solvent, which is what dissolving means. In a solution, which is what I've drawn here, recall that the substance that dissolves is the solute. So in this case, salt, for example. The substance that does the dissolving, which is almost always water, is the solvent. Water can also dissociate into its components, hydrogen positive and OH negative. The measurement of the amount of hydrogen in solution is what we call pH. This is measured on a pH scale. A pH scale goes from 0 to 14. 7 is neutral. 0 to 7 is acidic. And greater than 7 to 14 is basic. Water is not an atom, but a molecule. The other molecules we spend some time discussing were what we call the organic molecules or compounds. And we just did this, or are doing this currently. Remember, we have carbohydrates, also known as sugars. We have fats, also known as lipids. We have proteins, and we have nucleic acids. Carbohydrates and fats give you energy. Proteins do a variety of things. We spent some time especially talking about enzymes. And nucleic acids like DNA govern your genetic inherited information. We discussed monomers and polymers. Remember, carbohydrates are made up of simple sugars or monosaccharides, which we bond together to make polysaccharides like starch. Fats are made of primarily the primary monomer is fatty acids. We put together 3 of them to make a triglyceride. And proteins are made of a different kind of acid, an amino acid, and we bond these together to make the protein or polypeptide. Nucleic acids, we did not discuss their monomer and polymer, so you shouldn't have to worry about them. To look over your notebook and to examine any diagram we had of a microscope. Here's my microscope. How's this? This is pretty good. Nice. What do you want to stage? A little base here. Some lenses. That's pretty good. Knob. You should be comfortable with the parts and the functions of those parts. Recall that in class we have a compound light microscope. There's an eyepiece lens here, which is for magnifying, and there's also objective lenses here, which are for magnifying. For focusing, you have two knobs, a coarse or rough adjustment knob, which you do not use under high power because you might damage the slide, and a fine adjustment for use under high power to just minimally change the focus of your specimen. These help to focus or make your specimen more clear. They do not magnify the image. Your other parts would include the under the stage, usually you have a diaphragm, which is like that. We talked about a pinwheel, which you have a pinhole there that you can change the diameter for, which will allow more or less light from the lamp below up through the stage. So the diaphragm controls the light. You have the base for support. You have the nose piece, which holds the objective lenses. You have a body tube. Most of the other parts are pretty self-explanatory. Recall that when you look at something under a microscope, it's upside down and backwards, besides being, of course, magnified and larger. And in order to determine the total magnification of a microscope, you take your eyepiece lens and you multiply its power by the objective lens. So in this example, the total magnification would be 400 times. Good luck.